Welcome everybody. Welcome everybody to our Launch and Learn series, where today we will be diving into preclinical studies. This is brought to you by the ASGCT Patient Education Committee. My name is Dr. Rachel Bailey, and I am an assistant professor at the UT Southwestern Medical University, where my lab focuses on the development of gene therapies for CNS disorders. Today, I will be introducing our two wonderful speakers, Dr. Allison Bradbury and Ms. Kimberly Nye, and I will be your moderator for our panel discussion at the end of our Lunch and Learn series. So as a reminder, throughout our presentations, please feel free to submit your questions to our chat box, and at the end of after our final speaker, we will have a panel discussion where we will bring those questions up. So I'd like to introduce our first speaker, Dr. Allison Bradbury, who is an assistant professor in the Department of Pediatrics at The Ohio State University and is a principal investigator in the Center for Gene Therapy at the Abigail Wexner Research Institute at Nationwide Children's Hospital. Dr. Bradbury earned her PhD in biomedical sciences from Auburn University, where she focused on development of AAV gene therapy for GM2 gangliosidosis, which are currently in human clinical trials. Her postdoctoral research was performed at the University of Pennsylvania, where she evaluated disease mechanisms and developed gene therapy approaches for Krabbe disease. Dr. Bradbury received an NIH Pathway to Independence Award and joined the Center for Gene Therapy at Nationwide Children's Hospital in January of 2022. So with that, I will turn it over to Dr. Bradbury to walk us through the preclinical process. Dr. Bradbury?
Thank you, Dr. Bailey. Can you see my slides okay?
Yes.
All right, thank you for that introduction from Dr. Bailey and from the ASGCT Patient Education Committee for allowing me to share today and our topic of diving into preclinical studies. So I'm going to walk us through the roadmap of proof of concept studies all the way to the clinic, which encompass the whole process of preclinical studies. So the first step in this roadmap is really understanding of disease mechanisms. We really need to know what is happening, the pathways that are implicated in a disease prior to developing a therapy. The next critical step is development and characterization of models. This can be in vitro or cell culture models and or in vivo or animal models of disease. Once we understand the mechanism of disease and have models, we can then work on developing therapies. These can be cell therapies, gene therapies, small molecule therapies. Today, I'm primarily going to focus on gene therapy. Once we have a therapy developed, we need to evaluate the efficacy of that therapy in our model systems. This is really trying to show that there is a benefit of the therapy in the disease of interest. Once we've shown benefit or efficacy in our disease models, we need to then ensure safety and tolerability in healthy models, because the first goal really is to do no harm. Once we've proven efficacy and safety, we can then advance a therapy to the clinic. So the first step in this long pathway is truly understanding the disease mechanism. So we need to look at the molecular changes that are occurring and how those impact protein structure and function, because this really drives what the therapeutic strategy is. So for instance, if we look at a molecular change or an alteration in the DNA or RNA, we could see here, for one example, if there's a mutation in the DNA, that it may result in a loss of function. This may look like an absent protein, a shortened protein, or degradation of a protein. In this instance, a gene replacement strategy may be the best approach, where you provide a healthy or wild type copy of a gene to compensate for the missing or lost function of a protein. In contrast, some mutations or alterations in DNA or RNA might result in gain of function, where you have an accumulation or too much of a protein, and this can result in toxicity. In this instance, a gene knockdown or silencing of a gene would be necessary to reduce the accumulation of a toxic protein. So these are quite contrasting approaches based on understanding the molecular change. You've also probably all heard of gene editing in the last decade, and this is using a sort of molecular scissors to correct a very specific mutation. And this type of technology can be employed for many types of mutations. For therapy development, I'm going to talk today specifically about adeno-associated virus gene therapy. However, there are numerous different types of vehicles to deliver a therapy. So there are other virus types such as lentiviruses, and there are other non-viral vehicles such as nanoparticles or exosomes. Today, I'm going to talk about adeno-associated virus because that's really become one of the gold standards in gene therapy. And that is because this virus is small and it's non-pathogenic, meaning most of us have been exposed to it and it does not make us sick. Another advantage of this virus is that it has very low integrating properties into our DNA. Some other types of viruses previously used in gene therapy could integrate into the DNA, and that can result in off-target effects such as cancer. So this really is a safer technology for virus delivery. However, over the years, many different types, known as serotypes, of AAV viruses have been identified. And so depending on the organ or tissue that is affected in a specific disease, you need to choose your different AAV serotype in your preclinical studies. So for example, if this is primarily a brain disease, you might want to choose an AAV that is considered AAV9. Compared to if muscle is the primary organ impacted by a disease, you might want to look at AAV6. For the eye, AAV1, and so forth with heart, lung, and liver. So you really need to understand all of the organs that are affected in a disease and which AAV virus types would be best. And in some preclinical studies, we need to compare multiple different serotypes to determine which is best for a given disease. And in some cases, some of the naturally occurring viruses are not the best for a specific organ or a specific disease. And there is a new technology that is starting to develop novel AAV capsids. Here, what is done in the laboratory is there is an AAV capsid library is generated, taking parts of many different types of AAV serotypes, and those are then injected into animal models. And so for this instance, if the brain is the organ of interest, you can inject the viruses and pull them out from an organ of tissue, or even getting more specific, a cell type of tissue, and find which properties of those viruses are best for that organ and even a specific cell type, and really then validate specific novel candidate capsids. One recent big success story with this is a new AAV called MyoAAV. This was developed from this exact concept I just described, where AAV libraries were combined and injected into mice and to non-human primates. And this novel virus, MyoAAV, was pulled out of muscles, and then it was treated into a mouse model of a muscle disease and shown to have really superior therapeutic efficacy as compared to AAV9, one of the more standard AAV serotypes. You can see here in this picture of a muscle where the MyoAAV, the new muscle tropic capsid driving a fluorescent protein, has much greater expression in the muscle as compared to AAV9 with the same protein. The goal here is if you can become more specific with your targeting of a tissue or organ, then you can use less of the vector and really enhance the safety profile by more specifically targeting for your disease and requiring fewer virus particles. If you think of the vector capsid as helping you to drive expression into a specific organ or tissue, you can even further refine the targeting of your virus by some of the internal components. So one of the primary internal components of your gene therapy vector is called the promoter. This is the part of the virus that drives the transgene or your gene of interest expression. And by altering the promoter, you can start to change the expression at the level of the cell. So for instance, if you're trying to target a disease that primarily affects the brain, then you might choose the AAV capsid of AAV9. But then if you want to target neurons within the brain, you might choose a promoter known as CBA. If you, however, are working on a demyelinating disease that primarily affects oligodendrocytes, then you might want to change that promoter to one called myelin basic protein to drive expression specifically into oligodendrocytes. If you're working on a disease that primarily impacts astrocytes, another type of cell in the brain, then you might want to, again, refine your promoter to an astrocyte known as GFAP, which is primarily found in astrocytes. And we can see that by changing the promoter, so here with a promoter that's targeting astrocytes, we can hit targeting oligodendrocytes, you can hit almost entirely oligodendrocytes. But when you change just the promoter of this vector, you shift the expression almost entirely to astrocytes. So again, in these preclinical studies, you might need to look at many different capsids to see what's best at targeting your tissue or organ of interest. And then you might also need to look at multiple promoters to see how you further refine the expression of your vector into different cell types within that organ. So again, this is multiple steps of optimization in the preclinical studies, depending on the disease mechanism. Once you've decided on your different AAV constructs, so your capsid and your internal components that you're going to use, you also need to decide how you're going to inject that gene therapy into your animal model. And this, again, is going to depend on the organ or the organ systems that are implicated in that disease. So for diseases that primarily affect the brain or spinal cord, we commonly deliver the viral particles into the cerebrospinal fluid or CSF, which is the fluid that bathes the brain and spinal cord, because this gets the highest number of viral particles directly to those tissues. In contrast, if you have peripheral organs, for example, the liver that need to be targeted, then you might want to deliver your viral particles systemically or directly into the blood system to target those organs. However, there are other organs that are difficult to target by CSF or systemic injection. These include the eye, the lung, specific parts of joints, and even muscles. So you can do direct organ-targeted delivery of gene therapy in some of those cases. In August, the ASGCT Lunch and Learn is going to be on routes of injection, so please tune in to learn more about this part of the preclinical study design. Once you've decided on your therapy, so you have your vector, including your capsid and the internal components such as your promoter and your gene, you know how you're going to inject your vector into your animal model, then you need to really determine what are your outcome measures in your model to determine if your therapy is working. And really here, you need to think of what's going to be clinically relevant for the disease that you're trying to treat. So for instance, if you have a disease that is primarily a seizure disorder, then you need to be able to determine in your model if you are able to correct that seizure phenotype that is known to happen in the patient population. So for example, we might perform EEG in a mouse model of a disease to closely mimic what is done in the human patients. If you have a disease that has a gait abnormality, then in your mouse model, you want to ensure that there's a gait phenotype and that you can determine in your model if you're able to improve that gait phenotype with your therapy, so you can ensure that this translates to the clinic and to the patient. Similarly, many disorders are diagnosed and tracked by MRI. In that case, you would want to determine in your animal disease model if you can track the disease progression and therapeutic efficacy, for example, determining brain architecture and signal intensity on MRI. And some disorders, there's a cognitive impairment that is most important. And in those cases, you need to ensure that you have a mouse model that has some cognitive impairment. And again, you can determine in that model if you're able to improve the impairment by determining which outcome measures you're testing in the model. So this all comes back to really designing your preclinical studies to be the most clinically relevant to ensure translation to the clinic. You might wonder, how do we get these mouse models and how do we know what they look like? One of the most common ways of obtaining a mouse model is through the International Mouse Phenotyping Consortium. This is a cohort of over 20 different institutes that are located globally, and they are working to build a comprehensive catalog of the mammalian genome. And they're doing this by systematically phenotyping 20,000 knockout mouse strains. So they're knocking out each gene and making a mouse strain. They currently have over 8,500 genes that have been knocked out and phenotyped. And when they phenotype the mouse, they look at each physiological system and provide data on that as well as expression. And all of this data is freely available to the scientific community. And then the mouse strains are available to be ordered by researchers. So if you have a disease of interest, you can go to this website and see if a knockout mouse has already been generated and studied. One recent example of the success of this was just published in Nature in February of this year, in which they were able to look at a number of hundreds of different mouse genes and functionally link them to cardiovascular disease. And they were able to show, by using this discovery platform of single gene knockouts from the IMPC, that over 700 gene deletions induced a cardiac phenotype in mice. They were able to use clinically relevant measures, including ECG and even micro CT, to look at the structure and function of these mouse hearts. And then they were able to do comprehensive translation to humans by looking at alignment of heart disease of patients in a NICU pediatric study that was really what they called bench to bassinet. And they were able to find association of 5 new genes that had structural defects in hearts of babies. So this phenotyping of mice kind of works 2 ways, where you might have a disease of interest and you can see if mouse already exists, but also by systematically knocking out every gene, we'll also be able to find new genes that are implicated in diseases that we previously did not know. However, approximately 20 to 25% of knockout mouse models are lethal. And that's because in most patients, the mutation is not a complete knockout. It's somewhere further along in the DNA, and some of the protein exists. It's not a complete knockout. So we can use gene editing technology to generate knock-in mouse models now, where you can choose a specific indel or mutation using gene editing and put those different mutations with gene editing into embryos, transfer those into mice, and end up creating a CRISPR gene edited mouse model. The main advantage of this really is that you can generate patient-specific mutations, and this is something that we're doing in some research programs in my lab, where we've had a specific patient mutation that we wanted to study, and we were able to work with Shanghai model organisms to generate that specific indel using gene editing and prove that we have carriers or heterozygous mice, which then we received in my lab, and we were able to breed them make affected mice study the disease progression in the context of that specific patient mutation, and then also test different therapies in that model and see how the therapies would respond to that specific mutation. So this is really getting to a level of precision medicine where you can look at a disease phenotype and response to a therapy in the context of a specific patient mutation. I will say that mice are not the only models of disease. Diseases are naturally occurring in large animal models, and we can also develop clinically relevant outcome measures in these larger animal models. So we have a Jacob sheep that has Tay-Sachs disease. And in this sheep, some researchers at University of Massachusetts were able to develop a cognitive test that included a maze. And they were able to determine that sheep affected with Tay-Sachs disease made more mistakes and took longer to get through the maze due to their cognitive deficits. However, when they were treated with AAV gene therapy, that was able to improve their cognition. There are many different other examples of large animal models of disease, including pigs, cats, and dogs. And that's because just as in people, mutations can arise in large animals, and it presents a pretty unique opportunity to study disease progression and therapies in the context of a larger animal. With any drug study, you need to perform dose response studies in the preclinical phase. Dose response studies determine at which dose you reach a therapeutic effect. So if you give too little of a drug, you will have a sub-therapeutic effect. Then you reach the therapeutic effect and have a window where you have the therapeutic index. However, as you continue to increase your dose, you will eventually reach a toxic effect. This is no different in gene therapy, where we need to determine what our therapeutic window is. And this was a very clear example in Rett syndrome when they evaluated gene therapy in the mouse model of Rett syndrome. With this gray bar here, which is a low dose, you can see that in the Rett syndrome mouse, when a low dose was given, there was no benefit on the increase of survival. However, when the dose was increased tenfold, the blue line, you can see there was clear therapeutic benefit with a significant increase in survival. However, when the dose was further increased, another tenfold, the mice actually died earlier than the mice that were untreated. So this shows there's a sub-therapeutic level, a therapeutic window, and then a toxic dose. So we really need to find our dosing window with gene therapy, just as we do with any other drug. Once you choose that dose that you think is your minimal efficacious dose in a mouse, we need to be able to ensure that we can scale that dose, because a mouse is certainly not a human. So how do we take the number of viral particles that we found efficacious in a mouse and scale that to a human? And often we use larger animals, including rats, pigs, and non-human primates. In addition to ensuring that we can scale our viral dose, we need to make sure of the immune response that occurs at that given dose in the context of these healthy wild type animals. We also need to evaluate biodistribution. So where did the virus and the protein of interest end up, which organs and cell types, and at what levels? And we also need to evaluate for potential toxicity. We know that when we give a virus by CSF delivery, we can get large amounts of particles in the dorsal root ganglion. So that's one area we pay special attention in toxicity studies. And when we give the virus systemically or into the blood system, we can get high number of viral particles which can cause toxicity in the liver. So that's another area where we pay special attention in these toxicity studies in healthy animals. When we talk about manufacturing of our viral particles, this is a quite detailed process, which goes from the development and production of a plasmid, which is the starting source material. You then have to expand the cells which the plasmid is transfected into. You then produce the viral vector, purify the viral vector, and then formulate and fill and do quality control of this virus. As you can imagine, this is a long and expensive process. So when we're first doing therapy development and efficacy and dosing studies, we use what's called research grade material for both our plasmid and starting material, and for our virus. However, before going to clinical trials, we need to use clinical grade source and vector material to ensure that it's made with the highest stringency of manufacturing. The FDA now requires that your clinical grade material be used in your safety and tolerability studies so that the same product that's going into people goes into the safety studies. We also need to then perform bridging studies between our research grade and clinical grade to make sure nothing has changed and how the virus will perform due to the changes in the manufacturing process between these two grades of material. There was an April Lunch and Learn specifically on manufacturing. So if this is interesting to you, I encourage you to go back and watch that one that is now available on demand. And lastly, we need to have interactions with the regulatory agencies which help guide the translation from preclinical studies to clinical studies. Typically what happens is when we have efficacy and dosing studies completed and we believe we can really show benefit of a therapy in our disease model, we have a pre-investigational new drug application meeting with the FDA. We share this efficacy data with them and see if they are in agreement that we should move this therapy to the clinic. At this point, we also share our study design of our safety and tolerability studies and get their feedback on if those studies are sufficient. Then we perform those safety and tolerability studies, taking into account the FDA feedback. At that point, once those are complete, we submit the investigational new drug application to the FDA, and then we submit an IRB, investigational review board application, to the institution that's going to be conducting the clinical trial. And only after both of those undergo extensive review and are approved can a clinical trial begin. And really this whole process of preclinical study design and execution needs to be patient-centered. So we need to be choosing in vitro and in vivo models that really recapitulate the human disease to the best possibility. We need to be developing a therapy, including the capsid and the internal components, just the promoter, the route of administration that's going to be best for the disease at hand. We then need to really ensure that this therapy is safe by conducting extensive safety studies and having close interactions with regulatory agencies to ensure they are in agreement with our efficacy and safety data. While all this is ongoing, there really needs to be concurrent clinical trial readiness, including a central registry so we know which patients are available and will benefit from our therapy, and also a natural history study so that there's baseline data on the disease progression so we can really know if our therapy has had an impact in the patients. And with that, I will just quickly acknowledge my lab team who works tirelessly on a number of preclinical studies. And I've just mentioned that the patients and their families that spend countless time and efforts providing samples and financial support to really drive these preclinical studies. They're really the ones that we couldn't do this without. And I've just listed a few of the foundations that I might have highlighted throughout my talk today. And at this point, I'll hand it back to Rachel to introduce the patient advocacy side with Kim.
Thank you so much, Dr. Bradbury, for that fantastic presentation and for walking us through the preclinical process and covering considerations and steps that researchers take when developing a gene therapy. And as a reminder, we invite the audience to submit questions or discussion points into the Zoom chat for us to cover during our panel discussion as time allows. So we will now pivot from the researcher's perspective to the patient advocate's perspective on this process. For this, it is my pleasure to introduce Ms. Kim Nye. Ms. Nye is the president and founder of Test Research Foundation, which is a nonprofit organization that aims to find better treatment options for SLC13A5 epilepsy. Ms. Nye holds a BA from Princeton University, and she lives in California with her husband and their 4 children, 2 of which have SLC13A5 epilepsy. So with that, I will turn it over to Ms. Nye to speak on the patient advocate's role in the preclinical process. Ms. Nye?
Thank you so much. I'm going to share my slides. Can you see those all right?
Yes, looks good.
Great. Okay, so as Rachel said, I'm Kim, and I'm the founder of Test Research Foundation, and I'm going to share the patient advocacy side of preclinical studies today. So like many of the people probably watching this webinar, I started our organization because my family is directly affected by genetic disease. My husband and I were young, recent college grads, and we had moved to Oxford, England for graduate school when our daughter Tessa was born in 2003. We thought we were having a healthy baby, but Tessa began having seizures shortly after birth and for no apparent reason. We saw her diagnosis shift from benign idiopathic neonatal seizures, meaning that doctors thought she would be okay, to catastrophic epilepsy, where we weren't sure she would survive until kindergarten. Medications, surgeries, diets, clinical trials, and research all failed Tessa for more than a decade. At this point, she was having hundreds of seizures a day, but we still had no idea why. I dropped out of graduate school so that Tessa and I could travel across the country and around the world to try to get her the best care. Fast forward quite a few years, and in August of 2013, our world came crashing down again when I gave birth to our fourth child, a little baby boy who we named Colton. He seemed healthy, but just like his big sister, Colton began having seizures shortly after birth. He was only hours old, but I knew my son would never talk or dress himself. Colton's birth made the medical puzzle a bit easier. A genetics researcher named Matthew Bainbridge was able to find a genetic marker for the disease my kids had, and that genetic marker is SLC13A5. But there was a big question at that point. Was this diagnosis the finish line and the end of our journey? Or was it the beginning of a new race toward better treatments and ultimately a cure? Our neurologist agreed that we should not stop with diagnosis, but instead we should use that diagnosis and try to translate it into better treatment options. And so in 2015, we started Test Research Foundation. We put up like a basic Facebook page and a basic website. And sure enough, I started to hear from more and more families across the country and around the world who had also received an SLC13A5 epilepsy diagnosis. And it started to become clear that we had a new disease on our hands with a consistent set of symptoms. So all of our children began seizing shortly after birth, and those seizures were really hard to stop with seizure medications. Our kids could barely speak, but they clearly understood a lot. And they all have a movement disorder. That means they walk very late in life, if they ever walk. Many are dependent on walkers or wheelchairs. The disorder affects both boys and girls of all races and ethnicities. And it has autosomal recessive inheritance. So many of the families had 2 or even 3 affected children. Because SLC13A5 epilepsy was a newly discovered disorder, this is what the landscape, the research landscape looked like when we started our organization. It was really just a bunch of zeros and nos. There were fewer than 10 kids who had been diagnosed, and there was no funding or research toolkit in place to accelerate the development of treatments. I knew that the future was going to be really bleak for my newborn son, and I didn't really see any hope in the research landscape. And so it became a question of how were we going to find the answers that we needed in the timeline that we needed them. And we really just rolled up our sleeves and set to work creating a research roadmap and really driving preclinical research. I'm going to zoom in on a few key topics that have helped us accelerate preclinical research. The first one is research conferences. So we've held 7 research conferences to date, basically 1 every year. And the first question is always, who do we invite? And for us in the early days, we started by looking at who was publishing on this disease or in this general disease space. And we invited those researchers to our conference. But it's important to invite a broad group of stakeholders. So also inviting clinicians, definitely inviting families, and including biopharma in the conversation as well. There are lots of ways to plan a conference, but for our organization, we pay for the travel and accommodation of our speakers and of the affected families. Sometimes we have small meetings, and those meetings are really powerful, and other times we have significantly larger meetings that are more open to the public. I really can't understate the importance of including and amplifying the patient voice. When we hosted our first research roundtable, it amazed me that the researcher who was studying the mouse version of our disease didn't know the researcher studying the fish version of our disease, and neither of those researchers had ever seen a child with the disease. And so it's led me to truly believe that patient-led organizations are uniquely positioned to build those bridges, connecting various research silos. And amplifying the patient voice also allows researchers to see the burden of the disease and to use this information to inform the research questions they're asking. Conferences also allow researchers to share their data more quickly than they can through publication. This data sharing accelerates research and, in our experience, really spurs collaboration. For our patient advocacy organization and for our scientific advisory board, conferences also provide a forum to examine where the gaps in the research toolkit are, and they've really helped us establish research priorities for the year and create a strategic plan for our research program. Conferences also help disease communities grow their research network. Lately, we've been putting a special emphasis on making sure we invite early career investigators, because those early career investigators seem to be the most likely to go on and continue studying our disease once they have their own labs and are farther along in their career. And they're lots of fun to have at conferences. Okay, the next topic I'm going to zoom in on is research funding. Money. Money is really difficult for most of us to raise as small nonprofit organizations. Most patient organizations are structured as 501c3 nonprofit organizations, but nonprofit is a tax status. It's not exactly a business strategy. And so strategically raising and dispersing funding is key. We view funding from Test Research Foundation as seed funding. And what I mean by seed funding is we give out small-ish amounts of money. It can be anywhere from $10,000 to $25,000 or sometimes even $100,000. But we view it as seed funding that allows researchers to gather preliminary data that is necessary to then apply for larger scale funding from other sources like NIH grants or industry sponsorship. We've also partnered with other rare disease organizations in order to maximize our funds and increase awareness. For instance, there's an organization called Uplifting Athletes that will match nonprofit funds for an early career investigator grant. And programs like Jumpstart have grants that help subsidize animal and cell model creation. We also apply for grants ourselves that help fund our staff and our programs. We received a truly transformative grant from the Chan Zuckerberg Initiative Rares One project, and that grant has allowed us to hire a scientific director and a development director. It also includes a lot of trainings that have taught us best practices and helped us create a strategic and operational plan. And we recently applied for our first PCORI grant, so cross your fingers for us. We've also gotten grants from biopharma for conferences. These are usually small grants, maybe like in the $1,000 to $10,000 range, but they're extremely helpful. And we recently received an NIH R13 grant for conference funding in conjunction with UT Southwestern. Good old-fashioned fundraising from friends and family has made so much of what the work we've done possible. I like to highlight to donors that the work we're all doing in this space is meaningful and impactful, and this is an immense opportunity to have curing a horrible disease be a part of our community and our donors' legacy. There internally for us, there are some really high risk but high reward projects that probably don't have other funding sources. It's unlikely that the NIH would fund some of the things that we think are really important for our disease community. And so we use these donations in order to make that work possible. And finally, academic and industry partnerships have been really meaningful for us. I'm going to delve into this more later, but in our experience, patient-led organizations are uniquely positioned to de-risk therapeutic development for industry partnerships. The next topic I'm going to zoom in on is disease models and the preclinical toolkit. Scientists can't work on a disease if they don't have disease models and tools to work with. But in my experience, there's not like one single model system that you can use to answer every question that needs answering. So for instance, a zebrafish may be useful for a small molecule drug screen, but it is not going to be particularly helpful for your gene therapy development. Mice are a really popular species in drug development, but even once you prioritize a certain species for a project, you'll have to have lots of conversations with your scientific advisory team and researchers about the specifics of that model system. So for instance, our research community now has a knockout mouse, a humanized mouse, some point mutation mice, and it's really because there's a certain question that requires a certain tool in order to answer it. As Dr. Bradbury mentioned, you can check out if there's a knockout mouse on IMPC, or the Jackson Laboratories is a group that a lot of nonprofit organizations work with to create model system, well, to create mice. And to get back to that question of why have we created so many different mice, or why have research teams created so many different mice for our disease? Usually we create a new model system because we are trying to create an animal with a stronger phenotype. So in our case, children with SLC13A5 epilepsy have a lot of seizures. However, we've had problems creating a mouse that has a strong neurological phenotype. And even if you can get a mouse that recapitulates the phenotype, we are humans, not mice. And so there are diseases that have been cured many times over in mice, but still have not been cured in humans. And so looking to other species and other systems in order to have a different model is really key. And that brings me to the idea of patient samples. Patient samples are an extremely important part of the preclinical toolkit. Patients can donate fibroblasts or blood to create iPSCs. I'll talk more about that in just 1 minute, but they can also donate blood to look for biomarkers, and scientists can turn patient samples into organoids, which may be especially useful for things like developing gene therapies. Patient organizations can also help with tools like antibodies and nanobodies. Registries and genetic reports from families can be used to examine specific gene variants and can help elucidate if there are genotype-phenotype correlations, meaning that certain variants on the gene result in certain symptoms. And it's really important for patient organizations to advocate for the preclinical toolkit to be made available to all qualified researchers. So biobanks, repositories, even CROs and core facilities, they can really help make sure that the tools that nonprofit organizations are funding aren't just for one lab, but instead are broadly available to all qualified researchers. Okay, so I thought it might be helpful to walk through one example of how our organization has helped create a new tool for SLC13A5 epilepsy. In this example, I'll be walking through the creation of patient-derived induced pluripotent blood cells, and that's often shortened to iPSC. So the first step is to strategically figure out which families have certain variants on their gene that are of interest to your scientific community. You want to make sure that you have like a broad group of variants, but also, possibly of different ethnicities or ages, or just as representative as you can get of your patient population. And then we really just reached out to those families and tried to empower them to participate in our research. And what that meant in this case was having the affected child or children's blood drawn, as well as the blood of one of the parents. So in our disease, parents are carriers, and being a carrier is actually really interesting to our research community. And so we did both the affected kids and at least one parent carrier, and we sent that blood to our research partner, who in this case was someone at a core facility, and she turned those blood samples into induced pluripotent stem cells. And she agreed, we talked about all of this ahead of time, but she agreed to ship them to all interested researchers. And we did have researchers who have requested those iPSCs. And those researchers are not even funded by our organization. They just said, oh, great, here's a wonderful tool that I can use in my lab, and I'm going to give it a go. But we also wanted to make sure that some of our priority areas were studied. And so we did also put out a research funding announcement in order to encourage the use of the cells. And we funded a research team. In our case, that team is in Austria, in order to help characterize the model and work towards creating a screenable assay. We also had separate researchers who said, these iPSCs are great, but we need more differentiated cells. And so our disease is a neurological disease. And so we had researchers who turned the iPSCs into more brain-like differentiated cells that could be neuroprogenitor cells, or even more differentiated cells. And those cells were then made available to all interested researchers as well. As time goes on and we learn more about this model system, we're hoping to use it in order to screen potential small molecules. So, medications that you'd likely take by mouth, and look for things like repurposed drugs that would help our community. The final topic I'm going to zoom in on is drug development. We all want better treatments for our diseases, and how we turn that 0 into a 1, or even better, a 2 or a 3, so that we don't have all of our eggs in one basket, is really challenging. Mostly because we have such small patient populations, but it's not hopeless. There are lots of things that we can do to really drive and accelerate research. And the first step for us was assessing the landscape and figuring out who the key players were in the gene therapy space. So when we started diving into gene therapy in about 2017, there were really only a handful of labs who were tackling gene replacement therapies for monogenic central nervous system diseases. And the next step for us, once we figured out sort of the who, was thinking about what potential therapy options were for our disease. So SLC13A5 epilepsy is a recessive loss of function disorder. And that means that an antisense oligonucleotide is probably not the best option for us, but a gene replacement therapy is. And we are fortunate that our gene fits on a currently available viral vector. When we were thinking about gene therapy, we were also thinking about regulators and what type of gene therapy was realistic to get into clinical trials in the next few years. Gene editing seemed to us like the timeline was probably farther in the future to get into clinical trials, whereas a gene replacement therapy with a AAV9 vector, which had already been used in other clinical trials and was sort of assessed to be safe, we thought that was probably our best bet for a good drug in the timeline that we really needed it for the kids who are sick now. This process of gene therapy drug development is really expensive. It's like probably $3 to $10 million for a disease like ours once you factor in manufacture and clinical trials. I hear more and more about families starting their own drug companies, but our strategy has been to de-risk drug development by funding proof of concept research in academic labs, and then allowing industry, or begging industry, to step in and move the drug toward clinical trials and approval. For instance, our SLC13A5 deficiency gene therapy was started in an academic lab at UT Southwestern, and it's now in the pipeline of a company, Taysha Gene Therapies. Similarly, those patient-derived iPSCs that I described described a few minutes ago. That started in an academic lab at IST Austria, but there is now a spin-off company called Neuralentech that is really spearheading the drug screening and discovery portion of using those iPSCs. Okay, number 4, clinical trial readiness. It's a really key thing for patient organizations in their efforts to accelerate research. Natural history studies, biomarkers, and outcome measures are all part of clinical trial readiness. For our disease, we actually have a 3-part natural history study and biomarker discovery study currently underway. The first part, I'm going to pop up some logos of the people we're working with. So the first part is a remote study with telehealth visits for mostly our international families who would have a hard time traveling to the United States to participate. And the second part is an in-person natural history study. And we have 3 sites for that portion of the study. So there's a site at Stanford, a site at UT Southwestern, and a site at Brown. And the third part, we actually partnered with a company called Citizen, and they do a electronic medical records collection and a retrospective natural history study. So we can see what the disease has looked like in the past, and then the in-person and remote studies will continue to follow what it looks like moving forward. These studies we consider complementary. We have them all linked, and we really hope that they will elucidate key biomarkers and inform clinical trial design, and ideally serve in the place of a placebo arm in a clinical trial. And finally, number 5, I just wanted to highlight some of these timelines and the idea that we're not waiting for the preclinical studies to be completed and then starting the patient-based studies. Instead, we're running both in parallel. So I highly encourage all the patient organizations and patient advocates out there to try to launch their patient-based studies concurrently with their preclinical studies, because one really does inform the other. And involving families from an early stage encourages patient-centric trial design, and the information really will inform the IND process in talks with the FDA. Okay, so where are we today? We've made a lot of progress over the last 7 years, and this is what our research landscape looks like currently. We're in the triple digits for patients diagnosed. We have over $10 million going to research on our disease. We have not only a patient registry, but also a natural history study, or a 3-part natural history study. We remain the only advocacy organization in the world dedicated to this disease. We've held 7 research conferences. We now have access to free and accessible genetic testing. We think we have some predictive biomarkers, and we have more than 14 different model systems. We have some treatment guidelines in process. Unfortunately, we still have no idea why a lack of citrate transport causes epilepsy. And although we don't yet have a disease-modifying treatment, we do have one gene therapy drug in development. This, I hope, has been an overview of how the patient organization can accelerate and drive preclinical studies. I've seen my kids, Tessa and Colton, have so many seizures, and I really hope that the next generation of kids have less. But what I do know is that there's no one more invested than all of us who are directly affected by the disease. So I thank our partners who make sure that a diagnosis of SLC13A5 epilepsy comes with a solid treatment plan. And with that, I'll hand it back to you, Rachel.
All right. Well, thank you so much, Ms. Nye, for sharing your powerful story and perspective on the patient advocate's role in the preclinical process. So I'll now invite both of our speakers to join short questions and answers sessions. And as a reminder, questions can be submitted through the Zoom chat or Q&A features. And so we have time for a few questions. So I'll actually start by posing a question to Dr. Bradbury. So one of the questions I most frequently hear is around the timing of the preclinical process. Can you talk about how long preclinical studies can take?
It's a very common and tough question, Dr. Bailey, and it really depends on where your starting off point is. So for some of the indications that we work on, We come in and there's already a well-characterized animal model that has a phenotype that recapitulates the disease. And that's, super helpful because we can basically start at the therapy development phase and test it in this already existing and shareable animal model. And for that, that really expedites the timeline. However, we have other diseases where patients are, getting picked up on whole genome sequencing. This gene that most people have never even heard of is, the culprit in this disease. And we're really starting back at that first step of my roadmap of what is the mechanism behind this disease? What pathways are implicated? What cell types are implicated? What organs are affected? And for those disease where we're, literally starting, at zero, it's unfortunately a much longer roadmap to get the basis of the disease mechanism. Then we have to generate the models, characterize the models, develop the therapy, test the therapy, and then do the efficacy testing. I would say in good case scenarios, it's really hard to put timelines on things, but 2 to 5 years is what I would probably say for the whole preclinical roadmap to be completed. And we've also had programs, where we get to those safety and toxicity and scalability studies, and some of them go, really seamless and we don't see any toxicity. And then we've had other safety studies where we actually, do see pathology or even clinical signs in the non-human primates. And at that point, we have to step all the way back to our vector design and maybe change some of the promoters or expression level, and then redo the efficacy studies and redo the safety studies. So it depends on one, where you're starting from in that roadmap, and two, if at any point you have to jump backwards to any of those steps to really make sure. I know everyone wants this to move, as fast as possible, and I understand that. And for most of these rare diseases, time is not on our side. However, at the end of the day, we really need to make sure that the best therapy and the safest therapy is what we move forward. And for that, we really have to do our due diligence at each step.
All right, excellent. All very good points. And then for Ms. Nye, what did you find as the biggest challenge during the preclinical process as a foundation trying to help it along?
For us, we really started with so little because it was a newly discovered disease. And so I think funding has always been a really tricky thing. When you're traditional fundraising, you reach out to your friends and your family and your network, but when you only have a handful of patients, that network isn't that big. And so it was really being diligent. We knew that diagnosis would be meaningful to families. In our case, this is a recessive disease, so it plays a role in everything from family planning, to actually helping with the care. We knew that the kids were likely struggling to find symptomatic care. So we made a big push for diagnosis. We got our gene added to epilepsy gene panels and really talk with the genetic testing companies. And that had like a ripple effect, right? So even if your problem is funding, the solution might actually be at the patient level of diagnosis. And that's where we pushed it.
Excellent. Thank you. And then also for Ms. Nye, hindsight is 2020. So if you could go back and change either the timelines or how your group contributed to the preclinical process for gene therapy for SLC13A5 epilepsy, is there anything you would change?
I think I learned a lot about like the grant cycle process. So in rare disease, it really is the arena of heroes. Like we really love our researchers and our research teams. And I think that's so important to have like a meaningful relationship with your network. And it's why, these researchers stick with your disease. But when it comes to funding, I think it really is important to be objective and to get, experts to review different funding requests and to make sure that you really are funding the best science and that you have people who can tell you, yes, this is an appropriate amount to be charging for this study, or this is an appropriate timeline, or this is unrealistic. Someone who can help you keep the research timelines and costs on track.
Okay, great. Thank you. So we are winding down on our time for the session. Again, I'd like to thank both our speakers, Dr. Bradbury and Ms. Nye. Both of you did a fantastic job of covering each of those perspectives. And thank you from myself to the attendees for listening in. And I will be handing it over now to Lexi Starosta so that she can give some closing reminders.
Thank you all to our moderator, Dr. Bailey, and our speakers, Dr. Bradbury and Ms. Kim Nye, for taking us through the preclinical process and sharing a little bit about how foundations can get involved in that. Thank you all, the audience, for attending. I hope you all enjoyed today's session. We will be posting this recording on Monday, so look out for the link in your email. It will be posted on the patient education website. So if you found this information useful, please feel free to share it with all of your networks. After this session, there will be an evaluation. So if you could please fill that out to give us your feedback, it'd be greatly appreciated. These sessions take place every single month. So join us next month, August 25th, where we will be talking about routes of administration. Dr. Kimberly Goodspeed will be taking us through concerns, limitations with the applications of different therapeutic delivery methods. We also discuss how these relate to delivery in animal models during the preclinical study and what routes are currently being used in approved therapies. Registration is now open on our website, so please visit that. And thank you all again for joining, and we hope to see you next time. Thank you so much.